peptide proteins from DNA, okay? Okay, the RNA structure, we kind of went through this yesterday, RNA and DNA, differences, right? One strand RNA, sugar is different, and the uracil, the base is different, okay? We got that, you should have this down by now. Also, location could be a thing, function could be another one, a couple other differences we added the other day. We'll recap those again tomorrow in its entirety. All right, so this is the kind of a new slide, and this will make more sense when we get into the rest of the translation. But these are the 3 types of RNA that you'll be responsible for. And so, you know, you already have mRNA done. We did that yesterday. So if you started that chart already, I kind of mentioned that, you might want to just go ahead and finish it now. So go ahead and write these down in this abbreviated form, okay? What you need to have. Some people did, some didn't. So let's make sure. So Emma, do we add a slide?
No.
Yesterday?
No, we didn't do the function.
You were just good. mRNA, function, basically carry the coded message, right? And it basically starts in the, you know, this starts in the transcription of the nucleus and goes out to the ribosome. So basically, what you need to know is mRNA is messenger RNA, what that really means. So it really plays a role in the entire protein synthesis. It basically brings a message, the transcribed message from DNA, and then brings it to the ribosome to be translated. So it's kind of the conduit between the 2 processes. Okay? tRNA is transfer. We're going to learn all about him today. He's called transfer RNA. What he does is he transfers the amino acids to the mRNA, and we'll see how that all works in a bit. And that's basically, they're just floating around out in the cytoplasm. They're out there already. They're looking for their mate, which is amino acid, binds amino acid to it, and it carries it to the corresponding place or codon on the mRNA. We're going to learn about that. That's translation. And the ribosome is a guy, the cytoprotein synthesis, it's a guy that facilitates all this. It's much more complicated than we're going to get into. There's 2 subunits, a large and a small. They're 2 pieces that come together and blah, 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 and do a bunch of stuff. But basically what you need to know is made of rRNA. So R stands for ribosomal, so they're appropriately named here, TR and M, for its function. So basically rRNA makes up ribosomes, okay? And that's really it. And again, you know that ribosome is a cytoprotein synthesis, so you're there. Yes, ma'am.
Do you need to draw what they look like?
No, and this is really not what you'll see in a bit. This is not really what T transfer RNA looks like. They always do this to make it look like a T for the name. And the ribosome is a big globby thing, and you can see that 1,000 different ways. You'll see in the video it's shown 3 different ways, right? So, nah, as long as you know. And the little handout I gave you kind of has all those on it too. We'll label that towards the end today to recap everything we were talking about today. All right, and here's a picture again that is kind of, you know, pictures worth 1,000 words. This is a 2-dimensional representation of the overall process, starting with transcription. We talked about that yesterday. And transcription, again, is just taking DNA through an enzyme called RNA polymerase and making mRNA. That's kind of it. And we talked about that yesterday, and it has some rules. Yes, ma'am?
Can you go back to the slide for a second and get it on?
Now remember, you're not going to have time to write every single word down, so abbreviate and/or this will be posted on the web tonight. So if you need to facilitate. All right. So basically, this is the same thing we did yesterday. Transcription DNA to mRNA. You should have that down. That should be like the first thing you have. This is a picture showing that happening. Again, remember the substitution of the T and U. T on DNA, U on RNA. Okay? So when this happens, RNA polymerase makes that change. And this is just another way of showing the same thing. RNA polymerase making the mRNA strand from the DNA strand. Okay? And this occurs in the nucleus. Is the nucleus. All right? All right, and remember when this happens, what you get is you get a coded message, and that coded message is in threes. So the mRNA, it doesn't show it like in threes like our little picture does, but it's read by the tRNA in what's called a 3-base pair codon. So the codon, again, is the 3 amino acid that base pairs that codes for a specific amino acid. I'm sorry, let me rephrase that. The codon is the 3-base pair that codes for a specific amino acid on the mRNA. Okay? And we'll see why that's important when we get to translation in a moment. All right, and then we talked about the universality of the codon. There are, in fact, a couple special codons. One is AUG, which is the start codon. It actually codes for an amino acid called methionine, and there's a lot of research in that department now. And then there's some stop codons, too, and you'll see that on your handout when we get to the code chart, okay? And this basically just talks about, again, the universality that all the organisms on the planet basically share the same codon, just like every organism shares the A to T to G to C, the universal rules that we've talked about before. Just make sure you kind of remember that part, okay? We talked about this again. What is it that takes the information from the nucleus? It's the mRNA. That's the guy who actually transfers that message from DNA out in the cytoplasm where it can be translated. And we did this yesterday, if you remember. If you have a sequence of DNA, you should be able to transcribe it using the base pairing rules. And we did this in class yesterday, okay? All right. Now, I don't think we did this yesterday in this class. Did we talk about junk DNA?
No.
All right, so this is new stuff. So get your pencil ready. Let's go. As we've discovered, a lot of errors occur in the process, and that should be pretty obvious because there's so many little pieces to all these processes, and so there's a lot of areas of where errors can occur. And so over time, even with these checkpoints we saw along the cell cycle and the repair mechanisms in the DNA synthesis, errors do accumulate. And so errors accumulate in the DNA and then are passed on to generation after generation after generation. And so what has been found is that if you take a strip of DNA, and which we've done, the Human Genome Project has basically covered this, you're going to find that there's areas that they call junk DNA. And what that junk means is that not all the DNA is actually transcribed into proteins. There's some of it that really has no seemingly function whatsoever. Okay? And so what is it? What is it? We'll talk about that in a second. It could be viral insertions that have occurred over our evolution of a species, mutations that have accumulated that just never really have gotten coded for. It might be a spare gene. In case one's broken, it can go grab a spare. A lot of research on that right now. Okay? So, scientists call those, thank you very much. I was thinking the same thing. All right? Scientists have names for this junk and the names for the good stuff. Okay? And so what you need to know is this, is the good stuff, the translatable material is what are called exons. Okay, I think these were named backwards personally. I think exons should be the part that are X out, but it's not. And I blame the guy who named these, okay? So the introns are the part that don't code. So if you can see this, if you're in the back, you might have a hard time seeing this. This is a strand of DNA, and it shows intron, exon, intron, exon. So the exons are the part of the DNA that actually holds useful information that when our mRNA is transcribed, are actually kept. So you've got little enzymes called spliceosomes. There's a whole bunch of them actually, and we're kind of grouping them all together here, that will go along and they'll read the DNA and they can recognize the junk from the non-junk. And what they do then is they come through the mRNA and they splice out the introns, get rid of them, okay? And then they splice the exons back together to make a usable piece of mRNA. So this is an additional step that happens to correct potential errors from being turned into something hideous in an individual, okay? So another step, another checkpoint in all these metabolic processes we study. And that's this whole idea of getting rid of the junk, okay? So what do I want you to know here? The fact that you realize that there's some DNA that not all DNA is basically has a purpose that we know about. And that DNA that had given a name is called intron. the DNA that we know has a function that's actually translated into a protein is called the exon, all right? And so we have a mechanism whereby we can recognize those during this process after transcription occurs. This is additional process to make sure that the errors are cut out and the good stuff is put back together, and that's what is actually goes on from this point on to be translated, okay? All right, ladies first.
So this stuff is all transcription?
Yep. Because you get this first and then you get that. So it's kind of a, yes?
Was there any that, if we wanted to, we could like change like how like newborn babies would look or something like that?
Well, yeah, that's actually a whole different thing. But if you, I think I see where you're going here. If somehow this overlaps into this, if somehow we, when that splicing is done, if it's done wrong and cuts into this junk, yeah, that's what, and this will get to the next slide, thanks for the segue. What is the purpose of this junk? Evolutionarily, it's thought that it might be an accumulation of mutations, which is possible. It could be accumulation of remember, viruses, when they come into your genome, they insert their own genome into it. So the accumulation of viral infections that have lasted and accumulated over time. Could be that it's, again, spare. Could be it's a raw material by which evolution can occur. Again, there's a myriad of hypotheses out there, and the answer to your question is maybe. Okay, how's that? Best like, and this is a huge, you can imagine, absolutely huge, very current area of research, because again, understanding if you can tweak that, yeah, you can find out all kinds of cool stuff. What is hidden in there? What else do we got that we don't know about? Is it good? Is it bad? Who knows? Okay. All right. Now, going back to the genetic code, just to recap, because we're going to use this a little bit, the genetic code again is this basically, and long ago they figured this out. All organisms share this, and basically these are the amino acids, and these are the codes, the codons that will specify when translated for the specific amino acids, with the idea that these are stop signals. And then you got a start signal over here. And the start signal manifests itself as a methionine. Methionine kind of acts like a start signal, or like a primer in DNA synthesis, okay? It is a start signal. So you'll see that in a second, how that all pans out. All right, so let's get into it. All right, translation. Next, second step in protein synthesis, which is take mRNA and makes a polypeptide / protein. Now, I said this once before. I'm going to reiterate it probably three times today. How these two terms correlate. Polypeptide is a generic term for many peptide bonds, meaning a bunch of amino acids. Okay, and in this process, one gene codes for one polypeptide, and many times that polypeptide is in of itself a functional protein. In fact, most of the time. However, as our technology got better, we found that in many cases there are many genes that are necessary for the formation of one functional gene. And so they revised the one gene, one protein hypothesis to say one gene, one polypeptide. So it's just a semantic thing here, okay? But for our purposes, make sure you know they're the same thing. And again, I'll show you in a bit the peptide bond that forms between the amino acids, so it'll kind of make sense to you a little bit, okay? But as far as we're concerned, same old, okay? Synonymous terms. All right, I'm going to, okay. Did you turn it off? All right, I'll turn it on in a second. Oh, you sure? That's okay. I'll delay for 30 seconds until we finish. All right, so let me go through the steps here, and again, it's going to be very difficult to capture all this in a two-dimensional form. And so as soon as I'm done with this, I'm going to actually show the video that shows this process again. and then we'll go back here again, and then we'll